Hello, my desk guardians. I hope you all are fine and enjoying a very safe and sound health. Today, we will talk about inflammation, its science. Although we talked about inflammation science in neoplasia video, but we will give quick overview about science too today. So today we will discuss about signs of inflammation, stimuli, goals, external as well as internal factors of inflammation, inflammatory components, and major steps involved in inflammation. So, without wasting our time, let's come towards today's topic, which is inflammation. And now we will see the signs of inflammation, which we discussed in neoplasia. So, as we know that inflammation has Latin origin, in rubor means redness, calor means heat, tumor or swelling, dolor or pain, and last one is result of all above signs, which is functionalism or temporary loss of function due to pain or swelling. Inflammation usually starts from a stimuli, for example, pathogen, which are common cause of inflammation. But inflammation can be caused by other stimuli as well, such as toxins or trauma. For example, after intense workout, your muscle can feel sore, that is inflammation, which is trying to repair your overused muscles. Now we talk about goal of inflammation. It is to respond to stimuli and restore your balance. Often it includes eliminating causes of tissue injury, clearing out necrotic cells, and starting tissue repair. Apart from these factors, inflammation can be triggered by external or internal factors. External factors can be non-microbial or microbial. Non-microbial factors include allergens, irritants, and toxic compounds. Now, two main microbial factors that trigger inflammation are virulence factors and pathogen-associated molecular patterns or PAMPs. Virulence factors are factors that help pathogens to colonize tissues and cause infection, while PAMPs are small molecules of conserved pattern that are shared across many different pathogens, including bacterial wall components like peptidoglycan, lipopolysaccharides or LPS, and lipoteichoic acid, and fungal wall components like mannan. PAMs may include viral DNA or RNA. Our immune system recognizes these virulence factors and PAMs and release inflammatory response against them. Now, in terms of internal factors, there are PAMPs, equivalent molecular factors, which are damage associated molecular patterns or DAMPs. DAMPs are intercellular proteins that are released when plasma membrane injured or cell dies. So, DAMPs are signals that there are serious cell damage and they trigger inflammation. PAMPs and DAMPs are recognized by pattern recognition receptors or PRRS that are present on surface of leukocytes that activate the cells and spark inflammatory response. Key factors of this response are non-specifics and super fast response, which occurs in minutes to hours, and finally there is no memory associated with innate responses. Generally, we know that there are two types of leukocytes, which are granulocytes having neutrophils, eosinophils, basophils, and mast cells, while agranulocytes have lymphocytes and monocytes. While monocytes are further divided into macrophage and dendritic cells, other inflammatory components are sentinel cells, which have macrophages, mast cells, and dendritic cells, and components like blood vessels, plasma proteins including complement proteins, antibodies, and kinins like bradykinins. Now we will talk about inflammation steps, and you can see that they all start with letter R, which are recognition, recruitment, removal, and repair. First step, recognition, occurs when sentinel cells and tissues recognizes DAMPs using scavenger receptors or PAMPs with TLR. After sentinel cells recognize PAMs using TLR or TAMP using scavenger receptors, they release tumor necrosis factor or TNF alpha and interleukin 1 or IL-1. TNF alpha and IL-1 act on endothelial cells and hence endothelial cell release adhesion protein molecule known as selectins. Now selectin will allow white blood cells to recruit through bloodstream to site of inflammation and in tissues. This process called recruitment, which is second step. It is important to note that TNF alpha and IL-1 can also travel to the brain where they act as pyrogens and induce fever. TNF alpha and IL-1 can also move to liver and stimulate it to produce acute phase reactants, especially MBL, which help to produce complement proteins called C3 in C3A and C3B form, which then undergoes opsonization and autolysis of membrane attack complexes. Complement proteins also induce degranulation of mast cells, resulting in release of histamine, which causes vasodilation and endothelial retraction. Endothelial retraction induces more leukocytes to reach at site of inflammation and allows plasma proteins like albumin to leave blood and enter the tissue. Now, albumin draws water into tissues to bring swelling or edema. Now, in a tissue recruited macrophages and damaged tissues begin to phagocytose pathogens and damaged cells. This is third step, which is removal, and as we know that final step of inflammation is repair. This is accomplished as white blood cells release cytokines and growth factors to initiate repair of damaged tissues. For example, cytokines release fibroblasts to lay down a new collagen matrix to repair basal membrane of epithelial barrier. So this was all about inflammation. In next video we will cover about acute and chronic inflammation. So see you all in the next video. Till then, keep smiling and don't forget to like, comment, subscribe and share with your friends. Thank you.